So today I would like to talk a bit about how we can study modifications and how we can start using this layer for clinical purposes. Although in our lab we work also on mRNA modifications and tRNA modifications, today I would like to focus on ribosomal RNA modifications. The ribosomal RNAs together with the ribosomal proteins constitute the ribosome, and basically we know that the ribosomes are not just this kind of machines that just decode the mRNAs and produce the proteins, but actually their structure can be fine-tuned to potentially achieve specific functions. So historically we thought about the ribosomes as this kind of constitutive and uniform entities, and now we know that this picture is rather limited, and there's many layers that we can actually add to this picture. So specifically, we know now that actually the ribosomes are heterogeneous at different levels. They can have different stoichiometry of their proteins. They can have different modifications in their RNA, and they basically can have all these different layers of variation, including different copies from the ribosomal RNAs in the genome, and so on and so forth. So for the talk of today, I just want to focus specifically on ribosomal RNA modifications. So how can we study ribosomal RNA modifications to start with? It's not very different from what there's in mRNA, just that there's many, many more modifications, and the diversity is also a bit larger in general. So most of the modifications are actually 2' methyls and pseudouridine, but there's actually a wide variety of others as shown here in both the 18 and the 28S molecules mainly. So how can we detect them? So we can basically use some methods that are related to Illumina, but usually those are targeted for one modification at a time, or we can now use direct RNA nanopore sequencing, which allows us to see ideally all the universe of different modification types. Even using direct RNA, we can study modifications using different types of algorithms. So at the beginning, we started by using basically errors from base calling. So you can basically distinguish two conditions, wild type and knockout, treated and treated, and then identify which positions were modified. Then the next generation of algorithms were basically relying on looking into the changes in the current intensity and other related features of the raw signal. And then by binning the reads into one type or the other, you could then even go to quantitative measurements. The third family of methods is actually base calling models, so modification-aware base calling models, because the default ones were modification-unaware, so you could only predict the 4 bases. Now with the new generation of algorithms, you can now predict more than 4 bases, including M6A or other ones. So we developed one some time ago, actually. It was recently published, but actually also you now have the ONT-based calling models available for a subset of modifications. But then in the case of ribosomal RNA modifications, the base calling models do not cover the majority of modifications. So how can we study them? So we have been using the first type of methods mainly. So using this the first type of methods, we have been exploring the mammalian heterogeneity. So I already somehow spoiled a bit telling you that it is heterogeneous, but how did we actually get into there? So what we started by doing was, okay, are modifications even different across tissues or during development? And what we saw by here looking into 4 different tissues and across 3 different developmental stages, we sequenced all these samples using nanopore sequencing, and then we identified differential base calling errors, which basically are all those points that are off the diagonal. So by looking at these positions, we then actually got a list of sites and looked into which of these positions are differential. And this is what we basically got. What you can see is that there's some specific modifications that seem to be, for example, only present in the embryonic samples, and some others actually get in the adult. More specifically, you also see tissue-specific differences. So in the case of the adult, there's many that are neuronal specific. So it seems that neurons in the adult specifically have a very different modification patterns than what we have, for example, in an embryonic liver from animals, in this case, mice. So, okay, this already demonstrated that at the descriptive level that these modifications are different. So let's just take a look at some examples, right? Because this was like, so I always like to see the individual examples, and also in this case, the cDNA, to demonstrate that these were not SNPs, actually, but rather true modifications. Because one of the things that surprised us is that we actually were seeing changing modifications at places that were not annotated to be modified, meaning that our reference databases covering the universe of modifications is actually very poor with what we actually find using nanopore sequencing. So the SNPs were discarded, but actually many of these sites were validated using orthogonal technologies. So for example, some of the two sites that actually are in the middle and were not labeled were identified later on to be pseudouridylated sites using nano-CMC, which means that we did chemical probing with CMC and then coupled to nanopore sequencing. You can also couple to Illumina, but for us it's easier and faster to couple it to nanopore sequencing. And they were validated basically to be actually pseudouridines. So this demonstrates that not only they are changing across stages in development, but also that many of them were actually unannotated. So we said, okay, so if they change during development, meaning that somehow embryonic stages are very different from adult stages, could this be also the case in cancer? Because somehow, like, cancer cells are like dedifferentiated cells. So for this, we actually chose 4 different tissues from human samples, and we then compared specifically the normal versus cancer for the same tissue, and did the same exact procedure. You can see that at least for this initial pilot experiment, both the lung and the testis seem to be different in the case of cancer. So we decided to explore this in a larger cohort. So for this, we collaborated with Karsten Muller-Tittel from the Heidelberg University and actually collected 150 samples from patients and sequenced both the normal and the tumor from the matched patients. And we then subjected this to nanopore direct RNA sequencing, and then we would do some training and testing later on to see how well we could predict whether a sample was coming from normal or tumor, actually. So this is actually the clustering of the samples. Each dot represents a sample. I would like to note that this is just ribosomal RNA modification profiles, right? We're not doing any deep sequencing of the mRNAs or anything related to that. This is just purely looking at the boring ribosomal RNA modifications that supposedly are uniform across people and across tissues, right? And you can see that already in the first principal component, you can already cluster the samples just based on, you don't need to do any fancy, trainings or anything. Already, the clustering in the first principal component shows that the samples are drastically different from the same individual patients, depending on whether you took the sample from the tumor side or from, the adjacent matched tissue. So this already was super encouraging, and actually the fact that this was so information-rich actually made us think, how low can we go for sequencing? How low would we be able to predict? So the question was not anymore whether we can predict, but actually, how low can we go? And this is just actually the random forest classifier that we ended up doing, where basically you still get very impressive area under the curve, even just from 100 reads, which somehow means that, like in blood samples, like plasma, for example, where you do expect to have very low RNA amounts, we do think that maybe we can already even do this from very low RNA input samples like plasma. And here, just in the right, I'm just showing some examples of these split in positions that are changing between the patient, right? So you can see, for example, that in the first case, there's a pseudouridine, that for most, but not all patients, it's increasing in one case, and in other cases, the modification is lost. So okay, how can we bring this to the clinic, right? So it seems that it's actually working. So what is the next steps to do this? I mean, the first thing that I did mention briefly, but it is an issue, is that actually you do have very low input samples typically, right? So you somehow start, for example, if you think about plasma, from 1 ml of material, and if you do an RNA extraction, no matter which kit you use, you're never going to get really more than 5 nanograms, right? So if you know the specifications of a flow cell, you actually need 1 microgram of total RNA. So this is actually about 200-fold less, or if you start from polyarylated material, it's 300 nanograms, right? So the solution that we somehow have been looking into is in using multiplexing. So multiplexing will not allow you to then recover the coverage that you need, but as I was showing you, maybe only need 100 reads per sample. So this is basically one of the reasons why we decided to retrain the models that we had been developing for the RNA-004 chemistry. So the algorithm was published recently, and then we now retrained the model for 96 barcodes for RNA-004. And basically, the performance is super high. We actually get 98% accuracy with 100% recovery. So if you're interested in this, get in contact. Ideally, we want to put it really available in GitHub super soon. The second issue is that actually the whole pipeline usually is a bit computationally intensive. So what we have been seeing is that in general, people, when you go to the clinic, they don't know how to, they don't have access to GPUs, or they don't have access to certain things that are more complicated, or to the pipelines. So somehow what people need is like to be able to hit the button and make the analysis very simple. And that's why what we have been trying to do is like an analysis while sequencing platform for ribosomal RNAs, and also in our case, we're also very interested in tRNAs as well. So the idea is to make this very simple, and the idea is that with a few molecules, maybe you're able to already classify your samples. So the idea is that you can now click this button, and the whole pipeline will be starting for you in a way that you can just already visualize your results. So now this is validated, but we're now trying to see if we can really bring this to a plasma sample. So these are our future steps. So with this, I hope that I have convinced you that actually ribosomal RNA modifications are very diverse. They are dynamic. They are not the same across cell types, tissues, development, or even disease. And that using techniques like direct RNA sequencing, we can really get this epitranscriptomic fingerprints to really get an idea of the status of your cell, or which type it is, or whether it comes from disease or not. So there's much more power into direct RNA sequencing than just looking into mRNA molecules. So with this, I would really like to thank the people from my lab, and also thank you for the opportunity to present our work. Thank you.